Okay, so non-Mendelian inheritance sources are in the mitochondria DNA, basically. That is independent of nucleus genes. So any specific trait that has been inherited in a non-Mendelian way originates in the mitochondria. That's the bottom line as for now. Um, we have organelles that can code actually for some of its mRNAs by itself, and but the rest originate from the nucleuses or nuclear DNA that is imported um, into the organelles. So that means that we're going to find mRNAs that are transported from the nucleus into organelles, but it doesn't mean that 100% of the mRNAs or the genes that are expressed are from outside of the organelle. Um, Such genes that originate outside of the nucleus are called extranuclear genes. Okay, so the genes that are expressed or that originate from the organelle itself are those extranucleus genes. Um, Cytoplasmic protein synthesis is the process of protein creation, right? In the cytoplasm. We've actually dealt with this numerous of times in CP636 Advanced Clinical Exercise Physiology and CP637, right? So we talked about that, and you know it's a major process of the cell, which you have a movie about that you're invited to see. So both transcription and translation of extranuclear genes are in the organelle itself. So everything happens there, right? Traits that originate from the cytoplasm synthesis are told to be of cytoplasmic inheritance. So it's just a game of words, right? Maternal inheritance is affected more at the ovum and not by sperm, meaning that there is a specific location where maternal inheritance gains its stronger influence. Since non-nuclear DNA is a bit different, it has a different duplication mechanism and rate. We're not going to go into the duplication mechanism too much. We're going to talk about it just from the outside, but it's not exactly the same as other genes, and it's duplicated according to the needs of the organelles in which it serves. So mitochondrial DNA, that is abbreviated as mtDNA, has more mutations and is more sensitive to changes in the population. You're welcome to see the movie. So the rate of mutation accumulation of mtDNA is about 10 times greater than nucleus DNA. That means about 2 to 4% per 1 million years, which is extremely high, by the way, relatively to what is known in genetics. The most common organization form of organelle genome is a single circular molecule with a unique sequence. The organelle usually has several copies of the genome. The genome varies in size, sometimes by more than one order of magnitude. So it's not identical, right? the ratio of nuclear DNA to mitochondrial DNA is about 100 to 1. So for every 100 molecules of nuclear DNA, we're going to find one mitochondrial DNA molecule. But every cell has more than one mitochondria, right? And thus it has multiple copies of mitochondrial DNA. Okay, so the more you multiply organelles in a cell, the more genome copies you have in that cell, right? So theoretically, if I start working out aerobically, and that induces an increase in the amount of mitochondria that I have, that automatically induces an increase in the number of copies of mtDNA that I have. We find that mtDNA represents function possibilities as well. So the mitochondrial DNA includes a number of protein coding genes that is small and does not correlate to genome size. So I'll just clarify that. A, mtDNA codes for proteins of function, but the number of genes does not correlate to our whole genome size. Makes sense. We're talking about one organelle within the cell and not the whole body, right? So basically, it codes for 13 proteins. You will find no introns and only exons. That also means, of course, from what we've learned in the past, that it will be a lot more collinear with proteins, right, than nucleus DNA. The number of tRNAs that are coded from mitochondrial DNAs varies from none to all. So it really depends on what cell we're looking at. The organization of mitochondrial DNA between organisms also varies. Some are extremely compact, and some have great variance in gene expression. And one of the known characteristics of mitochondrial DNA is that some of the genes overlap, and in percentage, it's a higher percentage of overlapping genes than we would find in nuclear genes. So what does mtDNA basically deal with? An extremely high percentage with cell respiration. Okay, so and all those complexes, the cell complexes that have a thing or two to do with cell respiration, are expressions of gene expressions of mtDNA in general. When we look at DNA replication, we see that there is a region called the D loop, and the D loop is a very simple way that assists with replication of DNA. So that's a very apparent and clear feature that you can find in mtDNA. When we look at yeast's mitochondrial DNA, we find that their mitochondrial DNA is actually bigger than humans. Then you can say, how does that work? We are bigger in size, so you might think that or expect that mtDNA for humans would be bigger. And their mtDNA actually codes for 25 different proteins. There have been found, 2 genes have been found in yeast that those 2 genes alone are as long as the whole human mtDNA, and they are related to cell respiration. The interpretation of this information is that yeast, this will sound a bit funny, but in a way they regard respiration, cell respiration, is more important than in humans. Of course, it doesn't mean that it's not important in humans and we will die without it, but the yeast cell have more abilities to conduct cell respiration. Some traits of the mitochondria are actually originate from the nucleus, and that raises a few questions. For instance, a couple of questions. Number one, is this part of normal evolution? If so, why and how? The second one is, number two is the mitochondria the result of what we call endosymbiosis, meaning that the mitochondria was a different organism that connected, was taken in to a second organism, and with time a relationship, a function developed. So it is believed that the human mitochondria has evolved through endosymbiosis, meaning taking in and living, finding a way to live with, incorporate with another organism. All right, you're referred to a movie, please see it. The bacteria that was brought into the cell in endosymbiosis, now the mitochondria, offered the host cell some kind of advanced aerobic abilities. This does not happen just because it would be nice, meaning that the incomer organism, in this case the bacteria that became our mitochondria, had to offer something new or better to the host. And in this case, it was aerobic abilities. The common ancestor of mitochondria, the chloroplast and Rickettsia bacteria, was a sort of a parasite bacteria. So it makes sense. The symbiosis process allowed with time for the now mitochondria to lose some of its functions because it does not depend on itself anymore. It has a host, and thus some of the genes were provided by the host. So that bacteria lost its independency by losing a few functional genes, and those genes have been offered and given by the host. The loss of the genes is assumed most likely to be metabolic pathways, were the ones that made the mitochondria, or that bacteria that became the mitochondria, completely dependent upon the host. It is possible that those genes were not lost, rather than just transported into the nucleus. So maybe some of the genes in the nucleus are the original genes of the bacteria that became the mitochondria. transfer of DNA into the nucleus is greater than into the mitochondria. So transfer of DNA between location exists, and it exists more in regards to transportation into the nucleus. Of course, in the course of evolution and in general, the ability to express transferred genes requires a change of the coding sequence. Of course, DNA has to physically be moved into the nucleus. That means that some kind of component has to link to that DNA and actually take it to another place. So nuclear proteins have been found to have a unique sequence, allowing them to be imported into the organelle. These are called leaders. So you need to have a leader in order to be moved into an organelle. When DNA is moved into an organelle, it is called post-translational membrane insertion. So first it's translated and then it's moved through the membrane. It cannot be moved before translation. And this process through evolution has a very good chance of altering the human race. Alright, so we finished chapter 3 of part 1. I hope you have enjoyed. Please communicate any questions that you have.